Good afternoon, and everyone who's joining us. Thank you so much for taking part in this session on the digitization of precision medicine. We've got a really incredible opportunity here to give you an idea of the scope of the work that's going on in the digitization of precision medicine work research in the country. I'm going to start off the discussion with a very brief 5-minute introduction into the idea and a little bit about some of the ideas where CSIR is getting involved in this area of research. And then we've got a number of speakers joining us from around the country who will be giving their own specialist sort of expertise in terms of different aspects of this. And so we look forward to those, and thank you to those panelists for joining us. We're then going to finish off with a short panel discussion, which we're quite excited about, and we look forward to your questions, which we'll be moderating. So precision medicine is a really exciting field. This session that we're going to talk about now is not the only topic where we actually get to discuss this. Any of you who were up at 8:20 this morning would have heard on SAFM Dr. Jerolen Naidoo, my colleague at the CSIR, being interviewed by Stephen Curtis on the microbiome mapping initiative, which he'll expand on more tomorrow. But there are also a number of other people presenting this afternoon, Dr. Stoychev, Dr. Govindarov, and Dr. Gumede, who all at the CSIR are doing different aspects of precision medicine work. So we look forward to those presentations. So I'm just going to start off with a really basic idea, and it's very simple. What is it? What is precision medicine? Why are we banging on about this concept? And a very broad idea is that it contrasts with this one-size-fits-all healthcare approach. And so traditionally we've imagined that in a particular patient population, and I'm going to use the example of different types of drugs for specific diseases, but with that treatment, if you give the entire population, and what we are using up until now, the standard approach of one drug, as in one treatment should fix all, it actually doesn't do that at all. We've got increasing ideas that as little as 20% of the population are going to benefit from something like that, if we create this idea that everyone is the same, and they're just not. Whereas, if we spend time, a little bit of time, working out which drugs work best for which different parts of the population, based on a number of different things. I'm going to use the theme of genomics in my particular presentation, but it could be proteomics, it could be microbiome, it could be specific cellular models of disease. We're actually going to get a lot closer to identifying a few more different treatments that are going to have an impact on the entire population. So it does make a lot of sense for us to pursue this as an approach. So I think one of the important things, and the problem with precision medicine, and I think a lot of us have this, is that how can we in South Africa or Africa even start using this concept of precision medicine? Isn't this something that only really advanced countries have the capability to do that? And I'm going to just explain to you briefly why it should be done here more so than anywhere else in the world. So the only way to really explain that from a genomics point of view is to go back about a few 100,000 years ago. Africa is the source of all Homo sapiens genetics, right? So all the Smarties, all the different colors originated here. And then about 100,000 years ago, a small group of individuals, a small portion of that population, then left Africa and populated the rest of the world. Now, basically anyone left of the Caucasus Mountains became the Caucasians, Europeans. And so what the problem with that is that for the most part, and this is a generalization, for the most part, drug trials are done on people of European descent. That means that from a perspective of drug treatments, we know that the drugs that are designed in clinical trials work on the blue Smarties, but we don't actually know what their impact is on the rest of the population. And if Africa holds the entire heterogeneity of that, this is where we should be thinking about drug development more so than anywhere else. So how does this impact healthcare in South Africa? And this is just the pipeline that the CSIR has been working on in terms of implementing and how we can contribute to what's more of a sort of low-hanging fruit of precision medicine. If you imagine that any one particular individual approaches their doctor and the doctor goes, well, for that particular disease, here's drug A, and the doctor prescribes them the current drug. The problem with that is that because it's not doesn't suit everyone, you have increased hospital stays. We know that as many as 1 in 12 people are being admitted to hospital, not because they're sick from the disease, but because they are sick from the drugs that they are taking. That's admissions. So then what happens is then it's a guessing game by the clinicians. Okay, well then we'll try drug B and then we'll try drug C. But we're not making intelligent decisions about this, and we could. And the impact of that in the public sector means that because there are no, and again, I'm using the theme of genomics here, but it applies to lots of different areas of information and research. It's because there's no specific genomics test done on the African population specifically, it actually is going to lead to increased fatalities. In addition to the huge hospitalization cost, which is the majority of the cost that is incurred by the Department of Health. If we look at the private healthcare system, we have this imbalance where some individuals of European descent might be able to send off for a genetic test overseas, but we don't have a fairness of a healthcare system in the private sector. So you've got people in the private sector, what do they want? They want to make sure that the people who are paying their stipends are healthy, get the right drugs, they get better as soon as possible. That's what's going to cost them less. So this is the sort of the current system. But this is where, if we brought in digitization, using precision medicine approaches, this is where we could be. Imagine now the doctor has multiple options of drugs, and then based on, and this is the digital health platform that we are embarking on at the CSIR, based on big data algorithms and intelligent information, we can actually give that clinician the opportunity to go, the best treatment, the best chance for this person is with drug A, not drug B. It's very simple. And what does that do? It means it's going to reduce the hospital burden that we see in the public sector. It's going to reduce the number of drugs that we play guessing work with. And it's going to create a democratization of healthcare in the country, because now everyone has an opportunity to get their best treatment outcome. It's very simple. That saves us money, and it creates a balance from a socioeconomic point of view. It's really not rocket science. So with that initial idea, I'm going to finish off, and I'm now going to ask our second speaker, Prof. Kotze from the University of Stellenbosch, to share her screen and introduce some of the ideas and some of the really exciting work that they're doing in their platform and how they're moving different aspects of precision medicine forward.
Thank you for the opportunity to give this talk. Can you see my full screen?
Prof, we can. Thank you.
Okay, so I was just that. Okay, so you can hear me well. Okay, so I've got 10 minutes. I will then shoot off. Thank you very much again. So this talk is really about how, obstacles that occur for medical scientists like myself. We want to see those changes that you mentioned. And then we know when we do research, we have to ask ourselves, what is it that we would really see happening, just like those things that you just mentioned? And then we really have to ask ourselves, but what are these obstacles in that path? And what tools do we have to overcome them? And do we have the tools? If not, we have to develop them. And once we have the tools, then we can't just leave it there and give it over to other people to, use it or find the best way to implement. We should actually, as scientists, and especially when we work in a medical faculty like myself and many of my students and colleagues, we should then use those tools in different environments to make sure that they are incorporated in clinical algorithms. So when we talk about, in the past, medicine was done many times. It was an art, it was an ad hoc, it was case studies. The doctors learned the experience, and then they moved. Presently, we still have, people are grouped into different classes, are classified, and that is really where it's now the evidence-based medicine, which is very good. But for personalized medicine, we have to move another step. We need to combine those two so that we collect actually a lot of information per individual. So currently, the status is still where, scientists would opt for, let's sequence 1,000 genomes. And that's also very important, but we need to take the next step to translate that information into individual patient reports. So this is exactly what I want to try and convey on this page. With the Medical Research Council, their very exciting development on the Genomics Center, and I found this photo on the right-hand side top from their website. I've been privileged as part of our research group to be one of those scientists invited to submit 3 of the 6 samples that was ever sequenced for the first time in South Africa using this most comprehensive genetic test, whole genome sequencing. And you can see there from Brigitte Glassman, and there was about 20 authors on the article, how high the quality scores came out for the entire whole genome sequencing read length. Then on the genomic side, this is a company that I've been involved in, just disclosed that for many years. And the reason for this company, and it's a spin-out company from the Medical Research Council, is to try and develop a research translation tool. So something that we could use to overcome these barriers. So it's exactly what Janine said. It is an integration tool. It makes these algorithms working very closely with doctors interested in genomics and how to integrate the genetics and the lifestyle factors and the medical drugs and so on. And what we use as the bridge between all these different disciplines is that we use the pathology as the bridge. So I'm currently working in the division of chemical pathology in the Department of Pathology at Stellenbosch University, and that was the right place for exploring this kind of interdisciplinary science. So for us to unlock Africa's diverse gene pool, which is the goal of this genomic center. So if we go back to my first question, what is it that we want to achieve? We really want to unlock Africa's gene pool. But we should then focus not only on one aspect of a disease. For each patient, we need to look at, is there something from this gene pool and tapering, and sort of going into a specific patient's gene pool that can be used to direct their therapy? Is there something that would be very, very useful for early intervention on a surgical level? And then once we have that information, which of those genetic variants that we can detect can be used to screen families for pre-symptomatic diagnosis? This is the power of genetics. It can actually prevent. It can prevent that a person do not respond well to a medication, pharmacogenomics, like you mentioned, and to prevent them from being hospitalized again. I mean, we all have the experience in our own families and people we know, where it's the treatment of the cancer or maybe another disease, and then the side effects happen and it's back and forth to the hospital. That is in the end where many of the costs are built up. And then intervention. A person can have cancer, for instance, but what genes do we have to check out early on, if we can, if we now have this whole genome sequencing, that might tell us that this person is at risk of a second cancer down the line. I mean, genes like BRCA1 and 2, TP53, they also contribute causally to the tumor pathology. And if we look at cancer, it's not one disease. Within a specific organ cancer, there's many subtypes, and each of them requires different treatment strategy. And I'm amazed to see how many articles are still published where people look at these single nucleotide polymorphisms or single nucleotide variants, depending on the terminology, penetrance and so on, that they would group 100,000 cancer patients together and expect to get, a reasonable answer. If you don't divide them initially into those groups that we already know from the pathology that they are different, and then look at the genetics, then you will not get an answer. You would get all these contradictory findings. So how do we apply genomic knowledge? I think this is what I was trying to get at, that we should not just look at it as one disease, non-communicable diseases and infectious diseases. We would first consider a way to present them rather to cure them. And there's lots of things that we can do today to make that change, to impact on it. Vaccines, family advice, like I said, which treatment, statins or lifestyle changes for someone with high cholesterol. If we know the genes that is contributing to that cholesterol, just like if we know the genes that contribute to different subtype of cancer histopathology that makes that it's not one disease but different subtypes, we can treat them much better. And then so we need to start looking at cancer also like that. And this is where our research projects come in, that we challenge that whole idea of just looking at cancer as one disease and trying to develop kits and tests that could help us to dissect those differences early on and then take it from there. So the challenge is really for us as scientists, as a medical scientist, I need these two things that I'm supposed to do. It is to develop a test that can give a result to a doctor where they could help with the patient's next step in their treatment or workup for the diagnosis. Or if it comes to the interpretation, how do I put all the little bits of pieces of information together, the pathology, genetics, lifestyle factors, what are all those things that can play a role into a specific pathway that could cause the disease that we are looking at? Because it's not just the genes, it is the genes that act within a pathway, and eventually it is the pathway influenced by many, many things that we have to target. So this is then the new development that has been developed with the Medical Research Council. It was a Newton Fund grant, and we were very excited about where we are in the process today. We really developed these test kits, starting with breast cancer, leading the way in personalized medicine, precision medicine also, where we could say, okay, what would be key genetic variants that we could place within very important control points in an algorithm or clinical pathway, where having that few snippets in someone's whole profile as a guiding point, where would the genetics add value? And that is how we should develop these point of care assays, so that they could just help us to get patients access into the whole scenario of genetic testing and genomic testing. Once you have that patient in the practice, and increasingly genetic counselors are now starting to look into using those point of care tests as a tool while they are doing counseling. This test can run on referral of a doctor, and by the end of the session they may be able to give a patient some indication. I don't say necessarily a final report, but just an indication of whether we can work with this result that we got. Is there anything that we can now do for this patient to treat them, or to advise on the family or change of medication? Or is it now that we have an uninformative result with this initial screening that we know is important in our population in this specific disease, and then need to move on? So then we can use as a next step, all these new advances in next generation and third generation sequencing like nanopore. And at the end of the spectrum, we have the whole genome sequencing that I mentioned from the Medical Research Council. So what are all those things between the extreme of the point of care on the one starting point as an entry level first tier test, and on the other extreme of whole genome sequencing, many other tests can be done in between. And when you really fail to come up with a solution for the patient, this is where we then can proceed to do whole genome sequencing, and that can be then very informative for patients. So what we've then developed, and this is now also part of the whole digital world, is to develop a multi-platform pathology support genetic testing framework. So there's different test platforms. The ParaDNA point of care is one platform. The whole genome sequencing and what comes in between, different platforms for testing. And also by doing that alongside your standard pathology, it is where the doctor would obtain much more confidence in using the results, because the one result would sort of support the other. And if you find something that really doesn't make sense, all along, you need to go back and see maybe there was a mistake or something you forgot, or you need to still do. So I hope that message can come through, because my biggest question with this research is, do we just jump from a genetic counseling done to whole genome sequencing, or is it really necessary for us to have these in-between tests? If you look now at this picture where we have a clinical interface, the Genomics database tool, that's the translation for reporting. If you look at familial, lifestyle, and the therapy aspects in research translation, is it really necessary for us to have this non-communicable diseases pathway report before we move on to whole exome or whole genome sequencing? I would say at this stage, it works very well for us. We have 3 phases. So the level 1 report would just be the questionnaire, where we look at all the aspects that we would need to know in order to properly interpret the results in the end. And that can already give you a report. I mean, if you have the height and weight of a patient, you can, in the report, you can you can, in your algorithm, you can change it into a BMI. If you know that this person's diet is lacking this or that food or so, and then you have a dietitian that works with you, and it translates into a score, and we published on it, and we know that what we see, the score is low, you will be significantly associated with higher cholesterol levels, or if the score is also very low for iron food, maybe, then you could expect that the person's iron levels would be very low. And then if you then test the genetic variants underpinning those abnormalities of biochemistry, and you could see, but it is a genetic contribution plus the environment. Obviously, you can change that environment and then monitor the patient to see if the change is actually making an impact. So this is the type of approach that we have to align these aspects to the pathology. And when the treatment, the pharmacogenomics is done, does the patient actually respond to this medication? Because even if the genes show that this is the indicated medication, we know it's never just one gene. There's other genes, you know, upstream or downstream that could affect it. And then we need to look further. And as we learn from each case, we would be able to improve the database, build the knowledge, and the next patient would benefit from that change that we have to build into our algorithms as we go along, case by case by case. So I really believe that by bringing in the point of care testing, this is the first in South Africa. There's lots of work for us to do with validation, the regulatory requirements. We're trying to figure all of this out along the way, but it is really to improve the access into the right care pathway, so that we don't just jump off and, you know, start screening people. We need to know what are we going to look for in that whole exome or whole genome sequencing. So this, I think my time is basically up, but this picture is just sort of showing a lifestyle triggered risk, therapy related risk, and family inherited risk. Those are all the things that we can look at if we ask the right questions from the start in the health questionnaire, reviewing the tumor pathology and chemical pathology reports, and rather take another week before you start, and then you are going in the right way. So I think my time is up, so I'll just quickly show. This is also how we look at it. You know, if you want an easy question, it's yes or no, but we know that nothing is so simple at different stages of, if we just take cancer as one example, different genetic tests would be applicable. But here is a nice picture of how we did whole genome sequencing and how we could even help with misclassification of variants by furthering it. So this is basically my last slide. We need to use a multidisciplinary approach. Here on this, we managed to land on the cover page of the South African Journal of Medical Science at some stage due to a PhD student, Kathy Grant, sitting there in the middle. Left to her is an oncologist and another oncologist, Rika Pina. The first one was Elizabeth Murray. There I am, histopathologist, Dr. Colleen Wright. You see, so it is really about how do you bring everybody together so that in future we can manage the patients not just on their germline, but also on their tumor, and how we integrate all of that. So thank you very much.
Thank you, Prof. That was great. I'm going to ask, we'll save questions for the panel session. I'm now going to ask Rizwana Mia from the SAMRC to share her screen. Rizwana, you have 10 minutes to present, and then one of our colleagues will send you a message when you've got 1 minute left on the Zoom call. Thank you.
Okay, thank you.
Thanks, Rizwana.
Janine, I appreciate this. So good afternoon, everybody. I hope you can see the big screen, full screen picture.
We can see you perfectly. Thanks, Rizwana.
Thanks. So I just want to start off, Janine gave a very, very succinct introduction on, what precision medicine is. And I want to basically talk about the South African Precision Medicine Program, give you an indication of what is it based on, where are we, where we want to go to as well, and a few salient things, bring everything together for context. So the South African Precision Medicine Program, and as you know, we based at the MRC. The MRC is mandated by the country to look at, the best health implementation strategies and solutions for our context. And our major strategy is based on our quadruple burden of disease. And we see the precision medicine program as something to look at this in a more sustainable manner. And especially in the area of non-communicable diseases, which is, we found, was highly understudied until we started investing in it in around about 2016, like major program investment into research in non-communicable diseases. And the precision medicine program is sort of based on that. And you know why we have to do this is because obviously, like Janine gave you a very good understanding of the practice of medicine and how it's changing. And so, how do we see this evolve and how do we see ourselves building it? And we know that it's based on a foundation of ethics and regulation and policy, and then key infrastructure that needs to be put in place. And of course, skilled people in the area of genomic science and data science and health systems. And we see these as the pillars of science that are underpinning the area of precision medicine, and for it to be enabled from a research and innovation perspective, because essentially that's where we're starting. And of course, this is all pulled together by patient data, patient samples. And so, around about 2016, when we started this initiative, 2015 or so, we initiated a South African landscape analysis. And our objective is to see the program ensure that there's a better health outcome for every patient. And we sort of broke this down, and we noticed that in this field, there are pockets of activity that is happening. And as a medical research council, we saw ourselves, you have to have a good vision and administrative leadership to create a program like this for the country. And we mandated and led by government. From a health infrastructure perspective, Janine gave you a very good perspective of the public and private healthcare. And so, there's this new initiative by the Department of Health called the National Health Insurance. And this is what's going to fund healthcare in the future. So this is very important, the spend in healthcare. And Janine also touched on that. So I'm not going to labor those points. The other area is obviously biobanking and associating phenotypic data with genotypic data and creating database and patient population groups that you can make inferences from. And this, of course, is required. You require integrated searchable health records. And if you look at South Africa, we have lots of activity in these areas. We have good infrastructure with this Center for High Performance Computing, and the CSIR plays a pivotal role in this in our data science arena. And we have the large square kilometer array program. We also house H3A Bionet and the H3A Africa program in from a data perspective in Cape Town under Professor Mulder. And so big data science is something that we need to harness and integrate with our health record system for us to offer better perspectives in this area. If you look at the MRC, we've also got a spun out company called Jembi, and you're going to hear from Chris just now, and they're playing a very pivotal role in ensuring that our public healthcare system is creating those digital records on our patients, and they're actively involved in that. And I hope Chris will touch on that. And then we have, the area of precise diagnosis. Now, around about this time, we didn't have key infrastructure offering whole genome sequencing in the country. So in order for us to create the knowledge on our populations, we need to have all of this type of infrastructure and localize this type of activity for us to build capacity and solutions. And so that's when the idea of the SAMRC Genomics Center basically emanated. And of course, we need to start actively involving ourselves with big pharma and looking at more precise drugs for our population. And Janine also touched on that area where there's not really much in terms of drugs available for the African population because it's highly understudied. And with all of this data, we're going to be able to create more applied and translational research. And we're going to rely mostly on our good science that's being done at our universities and our science councils, especially like the CSIR, that is seeing themselves also lead in this area. And of course, when you talk about integration of this data knowledge to targeted interventions, you heard from Maritha, and there are a number of role players that are involved in this, and Maritha is one of those very key people who has been very passionate about this, and you can see through her initiatives with genomics that stem quite a number of years that she's been trying to advocate for this type of methodology in treatment. And of course, this all is hinged through innovation and breakthrough technologies. And we have a very good scientific base in this country, which we need to leverage off. And but we do have very few in the area of genetics, in the area of bioinformatics. So there's a whole talent pool that has to be developed. So there's a lot of work and there's a lot of gaps that need to be addressed. So essentially our precision medicine strategy is meant to address these gaps across that innovation value chain. And we at MRC, we look for strategic partnerships, and we want to ensure we can start initiatives around creating harmonized data science initiatives, creating the momentum and skills in genomics and data science to drive precision medicine, empower our local ecosystem to create and innovate new knowledge on the African genome, and strengthen capacity development to feed downstream into economic development, to innovate new tools and solutions for the healthcare system, because we have to look at this in a full sustainable way. And how do we do it? Well, we at the MRC, we use what we call a leverage funding partnership model. And we know we receive a parliamentary grant each year, and we report to the National Department of Health. We've created something with the Department of Science and Innovation called the Strategic Health Innovation Partnership, which is basically a product development partnership model, looking at investing in opportunities that are mainly in the applied science to pre-commercial area. And we basically receive funds from the Department of Science and Innovation and the Department of Health to actually fund healthcare technologies, and specifically addressing product development or product development research. And Maritha's a very good example of how she has developed her products or her service-orientated model through a SHIP funding. And so the SHIP strategy is based on global product development partnership model, but it's applied in an African context. So, what have we done so far in precision medicine? If I think about it, we've now to date have invested 212 million rand. And this may not be a lot for international funders, but this is a lot for South Africa. So we started off in 2016 with a leverage partnership model with the GSK and UKMRC through the Newton Fund, where we had a pool of 111 million rand, and we invested in 7 projects focusing on host genetics and other causes in the areas of cancer, cardiovascular disease, and diabetes within the South African population. And these projects are coming almost coming to an end now. And then in 2017, we created a partnership with Innovate UK and the Newton Fund again, and we placed a very small funding pool towards more precision medicine tools and solutions, looking at product development and partnering with UK companies. And Maritha's pathology, her para-DNA test that she has developed and is looking at that, integrating that model into her precision medicine approach towards breast cancer testing is an example of a project that was funded under this program. And basically, we've got 3 product development projects there. 2 of those products are moving into the validation phase and local manufacturing, which is amazing. And then in 2018, I'll talk about the MRC partnership with the Beijing Genomics Institute, where we launched a high throughput sequencing laboratory to create large scale genome sequencing for the country. And we have a pilot project to create that momentum of genomic sequencing in that country, in that lab right now. And in 2019, we just recently created a partnership with Diplomics and invested in 10 pharmacogenomic projects addressing treatment failure and host characteristics. And we have, 2 projects looking at host characteristics of COVID-19. And of course, through our plethora of funding, We also, like Maritha said, this predates the program, but basically the spin-off Genomics and Jembi were funded by the MRC, but not under a precision medicine gaze as such. So I just want, everybody's very interested, always interested in the Genomics Center partnership with the BGI. And so I thought, let me just highlight that, but also more so highlight that, this center will enable us to run sequencing at scale, create that niche knowledge on the African genome. And it is there as a national platform to be leveraged and utilized, and plugs a very niche infrastructure gap in the country. So we don't have to have our samples leave the country anymore. It can be sequenced within the country, and the data generated and housed at the MRC. And the MRC has invested in large scale storage capacity for this data. And a very interesting thing about our genomic data is that it's not really, we don't really house genomic data in the country for publication like when you need to publish, a publication authority will authorize your login of your data. And we don't have a recognized archive in the country. And so this is a very big gap that we need to plug now. That's the next step. And we need to look at a more sustainable solution to doing that, because obviously, we want to conglomerate our genomic data and create the patient population data so that we can make inferences and create better tools and solutions for our people. And I just want to also touch on, maybe there's other granting opportunities that we're looking at in data science, and you'll notice that there's a new one by the Common Fund, and this is an NIH opportunity. I mean, this was not drafted by us at the MRC, but the DSI have played a very big role. The Department of Science and Innovation of the country have played a very big role in looking at the scope of this grant that's been advertised, and it closes on the 3rd of December. So this is a very nice opportunity to also maybe look at precisely that area of harmonizing data and creating those large data pools in order for us to stimulate this area of data science in precision medicine. And, just to conclude, I mean, at MRC will continue in terms of, putting up a partnership model to create momentum in genomic science in South Africa. We see ourselves as currently laying that foundation to enable a framework strategy for precision medicine research and innovation. And we're so proud that we've just entered into a partnership with the European Union and other African countries to look at integration of personalized medicine initiatives between Africa and Europe. And I'll probably be contacting most of you in a few months' time to participate in this initiative, where we're trying to understand, what would make sense from an African context in terms of creating and building on the program for us to advance in this area of personalized precision medicine. Thank you.
Thank you so much, Rizwana. That's fantastic and very exciting, and just great to see how much the SAMRC is supporting this. I'm going to now ask Dr. Chris Seebregts to share his screen to give his presentation. Chris, if I can ask you to keep to your 10 minutes so we've got some time. There'll be a message coming through from one of our colleagues. He'll send you a message 1 minute before the end of your time. Thank you.
Great. Thank you very much, Janine. And thank you to the CSIR for the invitation to present today. Can I just check that my screen is showing at the moment?
It's perfect, Chris. Thank you.
Okay, thank you very much again. As Janine said, my name is Chris Seebregts. I'm the CEO of Jembi Health Systems, NPC, which is, as Rizwana said, a spin-out company of the Medical Research Council. And I'm also the program director for the SAMRC Jembi Collaborating Center for Digital Health Innovation. So today I'm going to be speaking about responsible big data management. Usually I'm on the other side of the fence talking about the exciting developments that were described by the two previous presenters in techniques creating data and creating big data and data science efforts. But it's an important topic as well, is to reflect on the consequences of big data and some of the constraints and the the responsible uses of big data. Oops. Great. So just in terms of a background, so what is big data? It's got a bit of a nebulous description, but it generally describes increases in the volume, velocity, and variety of data, and I'll explain that a bit more on the next slide. It's generally been enabled by exponential increases in storage and processing power of computers, which has given rise to just a complete explosion of data, as well as other initiatives, as has been mentioned, data science, as well as Internet of Things, and various other initiatives that are steadily digitizing our world. So data science as well has emerged as a powerful way to combine data sets. So it's not just an increase in the volume of data, but also a sort of combinatorial explosion of linkage of data sets to create powerful new ways to visualize data, as well as simultaneously increasing the ability to analyze and interpret those data. So all of this is growing up at the same time, giving rise to new tools to extract meaning from big data and to generate hypotheses. So as I mentioned, big data describes increases in 3 elements of data, and they're various, as I mentioned, descriptions or definitions of big data, but this is probably distilling out the essence of it. So it's a large increase in the volume, just the sheer size and quantity of data that is being stored. For example, large genomic data sets, omics generally, images, and a whole range of other data. Velocity is the speed at which data is required. So there's also been a huge increase in the acquisition of real-time data from the real-time systems. That includes both conventional systems as well as the large increase in data being collected by mobiles and other devices such as wearables. And then also the increase in the variety of data. So different data types, for example, omics data, which is giving rise to a lot of unstructured data, as well as the other unconventional sources of data, both individual and combined. And we could think just of social media data sets and the public health data sets that Rizwana was mentioning earlier is a particular interest of ours. So sources for big data in healthcare include, increasing hospital record systems, medical records, registers, laboratories results, etc. As I mentioned, these are being largely digitized, steadily giving rise to increases in the size of those data sets as well as the linkages between them. I mentioned digital epidemiology from mobile devices and online data sharing platforms. There's been quite a significant rise in open science platforms, which have been being populated increasingly with open data sets, also being used for both in and of themselves, but also in combination with other data sets, bespoke data sets. So accessing and combining these open science data sets using data science techniques is also giving rise to new data sets. Also translational research platforms, sorry, that's off the screen there, but integrating clinical and omics data, giving rise to hypothesis generation aligned with big data and machine learning. So some of the constraints on big data in health are, that medical institutions are really only custodians of the patient data, and that the patient data, of course, remains the property of the patient, and that consent is required for secondary use. So, and this is not always taken into account when the data sets are collected. And person linkage is used to combine data and to create these longitudinal records. And apologies, this is off the screen here, but there's a common truism in responsible data use that data can be either useful or perfectly anonymous, but never both. So that gives voice to a common trade-off that we deal with in big data and responsible data use, which is that the more identified the data is, the more useful it is from a big data and data science perspective. But of course, that gives rise to various ethical dilemmas. So some of the traditional ethical concerns are that compliance with the law does not always guarantee that data is used in morally acceptable ways, or that public trust is secured. So we have various laws that have been promulgated that seek to protect patient identified data or person identified data, but they don't always guarantee that the data is used in a morally acceptable way. And known issues with big data and analytics include informed consent, privacy, confidentiality, diversity, data ownership, digital divides, collective rights, and the inclusive governance of research data. And some of the current ethical concerns that are emerging is that ethical aspects of big data are shaped by concerns regarding the social consequences of both the widespread adoption of machine learning and the ways in which biases in data can be replicated and perpetuated. So, as I mentioned earlier, with the rise in big data and with the combination of data sets, there's been a consequent rise also in the application of machine learning and other statistical methods for the analysis of these data sets. And these can both replicate and perpetuate and even accentuate some of the biases that are inherent in data sets. So as I mentioned, in terms of the legal protection, it's increasingly used to obtain access to data sets. So we see the rise of data use agreements specifically are being used to gain access to data sets, whereas laws like the POPI Act in South Africa and GDPR in the UK are out there protecting the use of personal data. And data use agreements, as I mentioned, are being used both for primary data sets and also for secondary use of data. Whereas at the same time, we have ethics committees and IRBs safeguarding the use of personal data for research. And sometimes we find that there's a tension between these two different worlds. So some of the funders have got requirements for making data open access as a requirement of the funding, which is not always consistent with the requirements of IRBs and ethics committees. So to some extent, these are still being worked out. So some of the, what I've termed the rules for responsible big data, which I've taken from this paper that was published a few years ago, are guidelines that can be used to help entrench responsible use of in big data research. And we see that some of these are being entrenched in codes of conduct that are being implemented by societies and by organizations working in big data research in healthcare. So the first one is to acknowledge that data are people and can do harm. So that's one of the most fundamental rules, is the recognition that most data represent or impact people, and simply starting with that assumption until proven otherwise places the difficulty of disassociating data from specific individuals front and center. So that's an important principle to bake into from the beginning. Then the recognition that privacy is more than just a binary value. It is important to recognize that privacy is also contextual, situational, and not reproducible. Just because something has been shared publicly does not mean that its subsequent use should be unproblematical. So it's not necessarily a simple, just it's confidential or not. Then the third principle is to guard against the re-identification of data. So this is another issue that has come up again with the increase in sheer volume of data sets, but also in computational power, is that it's becoming increasingly possible to re-identify data that was previously thought to have been de-identified. So it's important to guard against that and to ensure that data that's collected in the first place is protected against re-identification. And then practice ethical data sharing. So for some projects, sharing data is an expectation of the human participants involved, and that's a key part of ethical research. For example, rare genetic disease research, but in other cases, it's not an expectation of the data that's being collected. And again, RBs are helping with the consent processes to guard against that. Then consider the strengths and limitations of your data. In order to do both accurate and responsible big data research, it's important to ground the data sets in their proper context, including the conflicts of interest. And context also affects every stage of research, from data acquisition to cleaning to interpretation of findings and dissemination of results. The next one is debate the tough ethical choices. So as I mentioned, there are a number of tough ethical trade-offs that need to be involved in big data research. Obviously, we understand that the power of big data research can often be baked into the ability to identify particular data sets, but the rights of the patient and the consent procedures that have been put in place need to be traded off against that. Then, as I mentioned, developing a code of conduct for an organization, research, community, or industry is also an important way that some of the safeguards are being put in place to entrench responsible big data research. And then designing data and systems for auditability. So assuming that the data and systems that are being collected and put in place at some stage will be audited is a good benchmark to work towards as one designs data collection and systems. And then engaging with the broader consequences of data and analysis practices. It's important to think beyond the traditional metrics for success, and how might big data research lessen the environmental impact of data analytics work. So as part of being part of the broader community and the understanding that a lot of the computational power that we use today has an environmental impact. So those are some of the rules that have been suggested for responsible big data research that we think would be useful, particularly in the healthcare domain. Thank you very much.
Chris, thank you very much. I think we've run out of time here. I just wanted to pivot one question to Jerolen Naidoo, my colleague here. Jerolen, what do you think is the most important aspect of digitization for precision medicine?
Hi, thanks. I think, and this is something that you touched on earlier, the greatest value of digitization lies really in the democratization of precision medicine technologies across all socioeconomic borders. And from an organizational perspective, I think for the CSIR, this has been something that has been really important in the way we've set about looking at our digital precision medicine platform. Because as an organization, our mandate, our goal is to improve the quality of life for all South Africans through technological innovation. And I think when we look at some of these cutting edge technologies, things like getting your tumor sequenced or having your cancer tested against drugs before you're given a treatment, these are really groundbreaking technologies, but they're also really expensive and inaccessible to everyone. And I think the value that digitization brings is that by digitizing this knowledge, the benefits of these cutting edge technologies can actually be shared by everyone. So a quick example would be, for example, if we think about someone in private medical aid, or who's able to actually afford or pay for these tests. So they're able to go on and get themselves sequenced, and they can reap the benefits of that. But that information then sits kind of in isolation within a single medical aid provider or a single doctor's office. Whereas that information could actually also be of use to doctors in the public health sector and to patients in the public health space. And digitization really is a tool that will allow us to share this knowledge and the benefits of it.
Quite right. I think unfortunately we've run out of time, so we're going to end there. But thank you very much for joining our session, and to all of our panelists.